Hello dear learner, in this session I will take you through basics of CRISPR-Cas. At the end of this session, a learner will be able to list down key components required by CRISPR-Cas for its action. A CRISPR-Cas, which is an acquired immune system from bacteria and archaebacteria. This fundamental system from bacteria have been exploited by genetic engineers as a tool for genome editing. We see some key discoveries related to it in chronological order. A CRISPR locus, which was identified from the E. coli for the very first time in the year 1987, whereas a better half of CRISPR, a Cas, which were identified in the year 2002. A role of CRISPR-Cas was described in the year 2007 that CRISPR-Cas helps in resisting a life cycle of bacteriophage. Whereas for the very first time in the year 2013, a role of CRISPR-Cas for the purpose of genome editing was published. The subsequent slides, I'll take you through some of the key components of CRISPR-Cas system. A CRISPR, which stands for a clustered, regularly interspaced short palindromic repeats. It simply means that there are palindromic sequences which are repeated several times. However, these repeats are separated from each other by spacer DNA. A CRISPR forms a particular locus. A length of a palindromic repeat varies in the range 21 base pair to 48 base pair. However, for a particular CRISPR locus, a length of a CRISPR palindrome is fixed. A CRISPR locus from a bacterial genome is always downstream to a leader region, and this leader region found to possess a promoter from whereby this CRISPR locus undergoes transcription. A Cas, which stands for CRISPR associated proteins, which forms a complex with CRISPR RNA and helps in targeting or degrading target DNA in a sequence specific manner. A spacer DNA is a DNA which separates a palindromic repeats in a CRISPR locus. This spacer DNA is believed to be acquired from invading bacteriophages or from other mobile elements. A length of a spacer DNA varies in the range 26 base pair to 72 base pair. A spacer sequence is present on a bacterial genome. A spacer sequence is very similar to a protospacer, only difference a protospacer is located on a bacteriophage or on a mobile element. It is from this protospacer sequence of a bacteriophage a spacer sequences are acquired by bacteria during a bacteriophage invading a bacterium. A CRISPR RNA, in short CRRNA, is formed on transcription of a CRISPR locus from leader region, whereas a transacting CRRNA or tracer RNA in short is a sequence which is independently transcribed from a tracer RNA gene, and this tracer RNA forms a complex heteroduplex with a CRISPR RNA that guides Cas proteins for its action, whereas a protospacer adjacent motif is a prior requirement for number of CRISPR-Cas for its function. Thank you so much. In my next session, I will take you through a types of CRISPR-Cas and mechanism of action of CRISPR-Cas proteins. Thank you so much.